Thank you very much. Thank you, Johan. That's a very nice introduction. And I'm delighted to be here in this august grouping. I think there's probably an inverse relationship between how scared you are in front of an audience with the size of the audience. So the smaller it gets, it's probably more scary. And you'd be scared both because of the excellence of the talks before you, but also the size of the cohorts are so impressive. The granularity of the data, all the data presented this morning just makes me think that the psoriatic arthritis world, where I work, is a good bit behind where you are. I think we have moved, though. Certainly 20 years ago, we were still in nappies. We have moved, I think, to being adolescents in comparison to psoriasis. And I think you'll find that in the next few years, in particular with investment in research into unmet needs in psoriatic arthritis, which is occurring both in Europe and in the United States, and I'll come back to the European effort later on. I think you'll find that the next 5 to 10 years we'll catch up pretty fast. So I'm looking forward to that. One of the things we need to do, obviously, is to develop a diagnostic test for psoriatic arthritis, because one of the things that I can't help but think when I see reference to psoriatic arthritis and the cohorts that you have is that most of the time you're referring to patients self-identifying as having psoriatic arthritis. And I know from studies both in our own department and elsewhere that if you're relying on self-assessment of psoriatic arthritis diagnosis or self-reporting of the diagnosis, that you're missing about 30% of cases. So we certainly need to work on that, and that's something we're doing. I'm going to move on with the talk because I could go on forever. So I'm here to talk today about where psoriatic arthritis is heading, and these are my disclosures, and there are two I would mention in particular. One is that I'm co-president of the group of GRAPPA. You're familiar with the name. And I'm co-president with April Armstrong, and that's sending out, I hope it's sending out an important message, because this is the first time we've had a dermatologist and a rheumatologist as co-president of GRAPPA, and it's trying to send out the message that we're trying to collaborate with each other. And I think that's the way forward, that we need to collaborate with each other. I'm delighted that there have been recent contact from the IPC to GRAPPA in relation to how can we build on a collaboration, because I think that's the only way we can move forward, is with a strong collaboration. The other thing, just to mention in disclosures, is that I am one of the coordinating partner. I'm part of UCD, which is the coordinating partner for the Innovative Medicines Initiative Consortium Hippocrates, which is one of those big consortia that has recently been funded, which is addressing unmet needs in psoriatic arthritis, and I'll talk about that later. So what I'm going to talk about today is a little bit about unmet needs. I'm going to talk about two competing theories relating to psoriatic arthritis pathogenesis. Be focusing on work that we've done on HLA alleles and haplotypes implicated in susceptibility to psoriatic arthritis, studies of CD8-positive T cells, and then some new concepts that are emerging relating to pathogenesis of spondyloarthropathy, and I'm talking not just about psoriatic arthritis, but the broader area of spondyloarthropathy, before finally telling you about Hippocrates. So this is psoriatic disease. It's a complex disease, heterogeneous in all sorts of ways, and it includes obviously psoriasis and nail disease, but also includes involvement of the joint or synovitis, enthesitis, dactylitis, spinal inflammation. You've got this tendency to both cause joint erosion and bony resorption on the one hand, but also you've got new bone formation being a feature in many of our patients as well. And then you've got the comorbidities. So it's quite a heterogeneous clinical disease. There have been recent significant advances, some of them perhaps not so recent, but important to mention nonetheless, the CASPAR classification criteria, because this has at least allowed us to have some uniformity about the inclusion of patients in studies and clinical trials, whereas we didn't have that before. So the CASPAR criteria have been particularly important. We also have composite outcome measures. Both the MDA and the PASDAS in particular are significant advances. We have an improved understanding of the genetic basis of the disease, and I'll go into that in a bit more detail, and significant advances in treatment, particularly targeting cytokines. And I think it's those advances in treatment. It came in a sense the wrong way around, but I think it's the advances in treatment that have been teaching us more about the disease and more about the pathogenesis. So it came perhaps in a reverse order, but all are contributing to significant outcomes in patient outcomes. So there are, however, ongoing unmet needs in psoriatic arthritis, and some of them you will be very familiar with, and these include the fact that we don't know how to identify psoriasis patients who are at risk for psoriatic arthritis, and if we could do that, you could potentially consider disease prevention studies, and this came up a little bit earlier on today, but there is, in fact, a disease prevention study either starting or about to start in the United States, I think using guselkumab in patients with psoriasis who appear to be at high risk, but I'm not sure how exactly you determine that yet. But they are going to go ahead with that study to see whether they can reduce the incidence or the development of psoriatic arthritis. The second unmet need relates to diagnosis. As mentioned, we don't have diagnostic criteria or laboratory tests to discriminate PSA from either rheumatoid arthritis or other forms of arthritis. We don't know how to predict outcome, in particular radiographic outcome, and a proportion of our patients, about 50%, will experience radiographic damage, and some of them will experience very severe radiographic damage. So if you could predict that, you could potentially prevent it. And the last unmet need relates to treatment, so lots of new treatments, but we really know no way of knowing which patient will respond to which treatment, and we spend a lot of time cycling from one therapy to the other in an effort to try to find something that works. So all of this is reflecting the problem that we poorly understand psoriatic arthritis pathogenesis, in particular the underlying explanation for disease heterogeneity. So there are essentially two major hypotheses circulating relating to psoriatic arthritis pathogenesis. They're not mutually exclusive, but they do present very different ideas about the initiation or causation of psoriatic arthritis. And the first you will have heard of perhaps from people like Dennis McGonigal in the UK, who has been talking about psoriatic arthritis as being an enthocele-based autoinflammatory disease. And the second is that psoriatic arthritis is an immune-based disease driven by HLA class I antigens presenting as yet unidentified peptides to CD8-positive T cells. So this is the paper published by Dennis McGonigal in The Lancet back in 1998. It was a paper where they hypothesized that the synovitis of spondyloarthropathy was secondary to the liberation of pro-inflammatory mediators from the enthesis, whereas in rheumatoid arthritis, the synovitis in rheumatoid arthritis was a primary event. So they suggested that the whole explanation of the disease stemmed from inflammation at the enthesis. And over the last 25 years since this paper has been published, Dennis has produced, and others as well, some very compelling papers that support this particular hypothesis. I won't go into it all, but they certainly have shown very nice anatomical studies and imaging studies that highlight the role of the enthesis in patients with psoriatic arthritis. However, it would appear that the enthesitis mechanism likely applies to only a minority of psoriatic arthritis patients because it's only in a minority where the patient will experience enthesitis occurring in somewhere between 30 and 40% of patients. So it certainly, I think, would be the view of many people that enthesitis, while important in some patients, does not explain the occurrence of psoriatic arthritis in the majority of cases that it occurs. So the other suggested hypothesis is that it's an immune-mediated disease. And when I started working at this now many years ago, it was known at the time that psoriatic arthritis was associated with MHC complex genes, although it was unclear as to which genes were important. It was also shown that in HIV disease that both psoriasis and psoriatic arthritis can occur more frequently and more severely, suggesting that CD8 positive T cells may be driving the disease and not CD4 positive T cells as in rheumatoid arthritis. And it was also observed that rheumatoid arthritis did not seem to occur in association with HIV disease. Psoriatic arthritis develops after syngeneic bone marrow transplantation, and that was shown. And there was also response to therapy directed at activated T cells. And these were the various features that suggested that it was immune-mediated disease when I started working at it. And we started looking at synovial fluid and peripheral blood from patients with psoriatic arthritis. And what we showed was that in psoriatic arthritis, that CD8 T cells predominated in the psoriatic arthritis synovial fluid with a reversal of the CD4-CD8 ratio that is found in rheumatoid arthritis. We also showed that these cells, the CD8 positive T cells, were both activated and mature, expressing CD45-RO and HLA-DR. So I was presenting this paper at the, what was called at the time, I've forgotten what it was called at the time, the ACR. And a chap comes up to the poster to discuss the poster with me, and we agreed to meet for coffee afterwards to go into it in more detail. And this guy was a chap called Bob Winchester. Bob, some of you may know, based at Columbia University in New York. He's now in his 80s. At the time, he was a very senior rheumatologist, and he had published the shared epitope hypothesis in rheumatoid arthritis. So he was a very respected and knowledgeable person to have coffee with. So I was delighted to meet with him. And this began a collaboration which went on for, it still goes on, but certainly was much more active over a 20-year period. So the kind of questions that we've been addressing, firstly, we decided to look at the HLA alleles and haplotypes implicated in susceptibility to psoriatic arthritis. And we asked the questions, what was the genetic relationship between psoriasis and psoriatic arthritis, whether psoriatic arthritis was genetically heterogeneous, and whether psoriatic arthritis genotypes were associated with different clinical phenotypes. So I'll go through, these are some studies that date back to 2012 and 2016, but I'll go through them nonetheless reasonably quickly. So the first part was to compare two psoriasis cohorts in a homogeneous Irish population. So the way this worked was we provided all the clinical material, and I had a number of PhD students who would travel over to Bob's lab and do the work over there. They'd actually live in Bob's apartment, 42nd floor on 5th Avenue, not 5th, 4th, I think. And he looked after them amazingly. And this went on for quite a number of years. So the psoriasis cohort were patients who had no features of psoriatic arthritis, spondylitis, or enthesitis, and each of these people had been assessed by a rheumatologist, and they were presenting to a dermatology clinic. The psoriatic arthritis cohort was presenting to a rheumatology clinic, and a diagnosis of psoriatic arthritis was obtained. So the question was whether the genotype of psoriatic arthritis was identical to that of psoriasis, and whether the psoriatic arthritis, whether the genotype of psoriatic arthritis was homogeneous. So what was known, of course, at the time was that in most studies, patients with psoriasis, about 60% of them expressed HLA-C0602. So the question was, does the distribution of C0602 and also of HLA-B57 in PSA parallel that found in psoriasis? So these were the results, and I don't know that there's a pointer here, but anyway, you can see there on the first panel on the left is the psoriatic arthritis patients. Then you've got psoriasis patients and then the controls. And you can see that in comparison between the psoriasis patients and the controls, that 57.5 or roughly 60% of the patients were C0602 positive. In the psoriatic arthritis cohort versus controls, the percentage who were C0602 positive was significantly reduced to 28.7%. Increased still over the controls, but significantly reduced compared to psoriasis. And the same was true of B57. So we concluded that we felt we could reject the hypothesis of genetic homogeneity of the psoriasis phenotype. And we went on to look to see whether or not there are other HLA-B alleles that are significantly increased in the Irish psoriatic arthritis cohort. So we looked at, I'll just show some of them here, B27, B39, B08, and B44. And you can see that the percentage of people who were B27, B39, or B08 positive in the psoriatic arthritis was significantly increased compared to controls, and also increased compared to the psoriasis patients. B44, in contrast, appeared to be protective, as it was significantly reduced compared to both controls and psoriasis. So we concluded that HLA-B27 and B39 were susceptibility alleles for psoriatic arthritis, but not high-risk alleles for psoriasis in the absence of arthritis, and that HLA-B0801 was associated with PSA susceptibility, but I didn't mention it, but appears to be somewhat protective for psoriasis. So in conclusion from this first study, we felt that the development of psoriatic arthritis predominantly occurred in individuals who inherited any of the following alleles or haplotypes. It's much less likely to develop in those who were HLA-B44 positive. And we came up with a new hypothesis that psoriasis was genetically heterogeneous, with the musculoskeletal cutaneous phenotype of psoriatic arthritis defined by several unrelated HLA alleles and haplotypes lacking a relationship to HLA-C0602. So these results were validated shortly after we published in 2012 by the group in Toronto. So they produced very similar results to what we published, which was great. So the second part then was, so different genetic susceptibility genes imply different disease mechanisms and possibly different clinical course and therapeutic responses. So the question was, is there a phenotypic difference? And the first thing we looked at was the time duration or time lag between the onset of psoriasis and the onset of psoriatic arthritis. So on the left-hand side there, you see the years between psoriasis and psoriatic arthritis in the HLA-C0602 patients. And on the right-hand side, it's the time gap between psoriasis and psoriatic arthritis in those who are either B27 or B39 positive. And essentially, they're quite different. So there is a time delay of up to a mean of 11 years between the onset of psoriasis and psoriatic arthritis and those who are HLA CO602 positive, and that's reduced considerably to 3.84 on average in those who are B27 or B39 positive. So it appeared that the time interval between psoriasis and psoriatic arthritis was a genetically determined quantitative trait. And then we looked at other features, and I'm not going to show them all, but one of the features we looked at was this symmetricality or asymmetricality that occurs at the sacroiliac joints in patients who have spinal involvement. So what's been observed over the years is that patients with psoriatic arthritis frequently have asymmetrical involvement. They may either have no involvement on one side and a lot on the other, or there may be quite a difference in the degree of involvement between the two sides. And what we showed, and not surprisingly, was that symmetrical sacroiliitis, which is what you find in ankylosing spondylitis, was indeed associated with B27 and the associated haplotypes, whereas asymmetrical sacroiliitis was not associated with B27, but was associated with B0801. So we came up with the idea that, and this tree in the middle here is a reminder that this hypothesis emerged in a discussion we had while walking through this forest. It's the tropical red forest along the northwest coast of the United States. And this is a tree which has a very strange phenotype associated with it, some growth on the side of it. But we came up with the idea that there were essentially three types of disease expression in psoriatic arthritis that was determined by genotype. So you have those who are HLA B57 or CO602 positive, which appears to be associated with early onset severe psoriasis, but late onset, low penetrance, milder musculoskeletal disease. In contrast, those who are B27 positive, this was associated with early onset musculoskeletal disease, more synchronous with the onset of skin disease, with symmetrical spinal involvement. It was also associated with enthesitis, dactylitis, and with arthritis mutilans, in particular when the patient was also expressing HLA-C0602. HLA-B08 appears to be associated more with synovial-based disease, with joint fusion, joint deformity, with asymmetrical spinal involvement, and also with dactylitis. So we then asked the question, if psoriatic arthritis is strongly associated with HLA class 1 genotypes, it made sense to further explore antigen presentation by CD8 positive T cells. And I'm going to very briefly just present some studies we did of the CD8 alpha beta T cell receptor repertoire in psoriatic arthritis. And just remember that these were studies conducted 10 or more years ago. So this was the first of the two studies. This was published in Journal of Immunology. The first author, Patrick Costello, spent probably the bones of 2 years living with Bob. He did some fantastic work, focusing in particular on peripheral blood and synovial fluid specimens, which we had from patients with psoriatic arthritis, and we shipped over to New York. So we were looking at the CD8 alpha beta T cell receptor in the synovial fluid, and what we showed that there was an average of 32 major oligoclonal expansions. There was a small number of expansions that were shared with peripheral blood, but most of the expansions were not seen in the peripheral blood, emphasizing the specificity of the events, clonal events within the inflamed joint. There were some oligoclonal expansions in the CD4 T cell repertoire, but they were fewer and smaller, and also largely restricted to the joints, in other words, not shared with peripheral blood. We did sequence a single CD8 oligoclonal expansion and revealed that to be composed of a few structurally related clones with beta chains that encoded identical or highly homologous CD3 motifs. But these motifs were not shared among patients, And we were unable to find evidence of antigen drive occurring in these specific clones. However, we concluded that the data supported a model of psoriatic arthritis inflammation involving extensive and selective antigen and likely autoantigen driven intra-articular CD8 in particular, but some CD4 T-cell clonal expansions. The second paper, where Shane Curran was the main author, Shane also spent time with Bob, and we were focusing in this particular paper not on synovial fluid, but on synovial tissue. And what we did was we collected peripheral blood and synovial tissue from patients before and after they commenced treatment with methotrexate. And what you can see here on the left-hand panel is that during active arthritis, the T-cell receptors were largely polyclonal, but that during methotrexate treatment, the T-cell receptors became much more oligoclonal. And I guess that what that suggested to me is that these oligoclonal T-cells were remaining within the joints of patients with treated disease who had responded well to treatment. And then if you were to stop the methotrexate, the disease would just come back up again. So we weren't getting rid of perhaps the disease pathogenic clones. And it suggested that during active disease, that we were getting a lot of non-specific T-cell infiltration into the joint. These were not driver clones. These were associated with inflammation, but not associated with driving disease. But certainly seeing evidence of perhaps much more oligoclonal expansion during treated synovial tissue. So we came up with this autoimmune hypothesis, and that was that HLA class I antigens recognize self-peptides derived from proteins found in endothelial sites, perhaps, or synovial sites. CD8 T-cell clones were specific for these self-peptide MHC complexes becoming activated, with the activated state perpetuated by the continual supply of self-peptides. So this is the classic explanation for autoimmune disease. And there was some support for this with some studies coming from King's College and Bruce Kirkham's team in London, who showed clonally driven CD8 population containing an enriched population of IL-17 expressing, producing CD8 positive T cells in the psoriatic arthritis synovial fluid, which was not seen in rheumatoid arthritis. So nothing much happened in the area for then a number of years until this paper appeared in, I think, 2020. And this was coming from Hossein al-Maswabi, who was working in Oxford. And he used a different approach, which wasn't available to us at the time, but in the intervening years, single-cell sequencing became available. And he showed data that was very similar to the data that we had previously published, revealing clonal expansions of pro-inflammatory synovial CD8 positive T cells, which you can see on the right in panel D, significantly elevated in the psoriatic arthritis patients and expressing tissue homing receptors. So there was a threefold expansion of these synovial fluid memory CD8 positive T cells, pronounced CD8 T cell clonal expansions. They showed transcriptome analysis, showed them to be express cycling, activation, tissue homing, and tissue residency markers. TCR comparison between patients suggested some clonal convergence. And they also showed that the chemokine receptor CXCR3 was upregulated, while two CXCR3 ligands were also elevated in psoriatic arthritis synovial fluid. So it certainly appeared to provide support for the suggestion that these CD8 T cells in psoriatic arthritis may be of importance. So the implications of these observations was that they were firstly consistent with Mullen Wright, identifying skin predominant, synovial predominant, and axial predominant psoriatic arthritis, each having different HLA associations. Since different HLA molecules bind different peptides, this suggested that distinct autoimmune responses drive the different phenotypes. These autoimmune responses may result in one or another cytokine dominating, as in the skin, meaning a differential treatment response may occur in different genetic subsets. And how these genotypes might result in a particular clinical phenotype is unclear, but work in cutaneous psoriasis from Jörg Prince's group confirms the proof of concept, whereby CD8-positive T cells have been shown to react against melanocytes, and these melanocytes then secrete psoriasis-promoting cytokines, specifically after the regulation of an autoantigenic melanocyte peptide processed by ERAP1 and presented by disease-associated MHC class HLA-C0602. So meanwhile, what's been happening in axial spondyloarthritis? And I'm going to go through this quite quickly, just to highlight two reviews that have summarized the work that's been going on in spondyloarthropathy, and in particular in ankylosing spondylitis in recent years, all suggesting that spondyloarthritis is not simply an autoinflammatory disease, but is also characterized by immunoregulatory disbalance. I would highlight just two particular papers, one from Bob Binman's group in Toronto. This was a small number of patients. Proteomic and transcriptomic analysis helped to identify a mature CD8 T cell subpopulation enriched in gut-associated trafficking molecules, which was elevated in ankylosing spondylitis synovial fluid. And these cells were reminiscent of TRM cells. This population, possibly gut-derived, appeared to have dual cytotoxic and regulatory profile. In a second paper, they looked at gut-derived CD8 TRM cells, which appeared to be expanded in peripheral blood and synovium of spondyloarthropathy patients. And it appeared that these TRM cells expressed the intestinal homing receptor alpha-4 beta-7, suggesting that they're of gut origin. So the data confirmed the expansion of TRM cells in spondyloarthropathy joints, providing evidence of expansion in peripheral blood and in gut with alpha-4 beta-7 expression by peripheral blood TRMs, supporting the recirculation of these cells from the gut to the peripheral joint and the inflamed joints, peripheral blood to the inflamed joints. So that leads us to a new development within EULAR, which is a group which is called MHC1-opathy, which has recently been set up. And there was a recent publication describing the purpose of the group. But it describes a family of inflammatory conditions with overlapping clinical manifestations and a strong genetic link to MHC1 antigen presentation pathway. And these diseases include both ankylosing spondylitis and psoriatic arthritis, Behçet's disease, psoriasis, and birdshot uveitis. So all have a strong association with MHC alleles and gene variants of the antigen processing aminopeptidase ERAP1 and ERAP2. The hope is to present new insights into the biology of these MHC1-opathies, and they feel that this will strongly advocate for disease overarching and integrated molecular and clinical investigations. And it's a collaboration between rheumatology, dermatology, ophthalmology, and also fundamental and translational researchers. So I think it's an exciting new development within the field. So just in conclusion, several unmet needs have been identified. Psoriasis is genetically heterogeneous with the musculoskeletal cutaneous phenotype defined by several unrelated HLA alleles and haplotypes. Different alleles and haplotypes are associated with different phenotypes, and synovial fluid T cells are clonally expanded, in particular the CD8 population, which is particularly found in the patients who are on treatment with methotrexate, with similar findings now emerging from spondyloarthropathy, ankylosing spondylitis, and other conditions. There's the emergence of the MHC1-opathy concept. So last couple of minutes just to mention the Hippocrates project. So this is, as mentioned, a large consortium across Europe, which includes 26 partners. The consortium is being led from UCD by Steve Pennington, my colleague there, Professor of Proteomics, and myself. And it includes 5 FPA partners. It includes strong patient participation from both GRAPPA, from EULAR-PARE, and from Europso. And it also includes a wider network, which I refer to there as the GRAPPA EU network. All of these centres would have a lot of experience with patients with psoriasis and psoriatic arthritis, and including them allows us to potentially access additional cohorts of patients with those conditions. The project started in July 2021, so it's going just over 2 years. The budget it's actually 27 partners, I see. That's because AbbVie have joined the consortium. The budget is 22.5 million with a duration of 5 years. A similar consortium has been set up in the United States. It's just been started under the AIM program. So that's just getting up and going with Chris Richland and Jose Scher involved in that one, and we certainly plan to collaborate closely with that group. So what are we doing? We're addressing the areas of unmet need, including the development of a diagnostic test, including the ability to predict which patients with psoriasis who will develop psoriatic arthritis. We want to predict damage progression, and we also want to look at treatment response. So the data, which will include clinical data, imaging data, multi-omic data, signal finding studies, will all be put together in Work Package 5, which is all about data integration and analysis. So we've just signed off on a consortium-wide data sharing agreement, which will allow all of these data to be shared on a single platform based at the SIB in Switzerland. I'm going to just give a shout out to one study within the consortium, which is of relevance to you all here, and that's the Hippocrates Prospective Observational Study, or the HPOS study, which is being led by Laura Coates in Oxford, and which is addressing the whole area of can we identify those at risk of developing psoriatic arthritis in order that we potentially can prevent it. So this is a patient-led, web-based, longitudinal study of 25,000 people with psoriasis across Europe. And I've listed some of the countries involved. There are more. The patients will enroll themselves. Consent will also be obtained remotely. And we will collect biosamples. Initially, we have funding to collect biosamples from 3,000 participants. But we are hoping to obtain additional funding to collect more samples and different types of samples from as many as the patients as possible. So this study is based on the concept that roughly 2.7% per annum will develop psoriatic arthritis. So the patients at baseline are not supposed to have a diagnosis of psoriatic arthritis. They have psoriasis. And we anticipate that 60 out of 25,000, 675 patients will develop psoriatic arthritis per year. So if we can identify risk factors, either clinical, molecular, or imaging risk factors which reliably detect which person will develop psoriatic arthritis, then we can begin to design and conduct studies aimed at preventing psoriatic arthritis. So we're not going to do any studies on prevention, but we're going to certainly consider the design of such studies in the future. So we're collecting information from patients with skin or nail psoriasis who are over 18 years of age, who do not have a diagnosis of psoriatic arthritis. They may have some symptoms. They may, on assessment, have some disease, but they don't have a formal diagnosis of psoriatic arthritis at baseline. We intend to follow the people prospectively every 6 months for the development of psoriatic arthritis until 2026, and hopefully further. And I've mentioned the blood sample. These are the data we'll be collecting. So demographic, lifestyle, data about psoriasis, including treatment, self-assessed PASI, questionnaires related to musculoskeletal involvement, the PEST, PSED for impact on life, and if PSED negative, patients will fill out a treatment satisfaction questionnaire. We will also provide participants with feedback on the scores and interpretation, and advice, if necessary, that they should seek a rheumatological opinion. So this platform has all been designed by a team at the University of Oxford, and significant input from patients into the design of this platform. So I would like to acknowledge that. I won't go through all of this, but just to say that the study has now launched in the UK, launched at the end of June. It's launched just in the last couple of weeks in Ireland. We're going through the translations which are required to launch this in other countries, but the rollout is expected to occur in other EU countries across Europe before the end of the year, and that certainly would be our target. And just to say that these patients are self-recruiting. The advertisement, if you like, will be online, but we're also asking our dermatological colleagues to provide psoriasis patients under their care with a flyer and encouragement to self-consent and sign up to this study, because I think it will be an important study, particularly when it comes to validation of any markers that we identify in smaller studies. It'll be very important to have this kind of size of study available for us to study. So with that, I'd like to thank everybody for listening to me. I just acknowledge some of my former mentors, my colleague Bob Winchester, the Hippocrates Consortium, and my colleague in Dublin on the bottom right, Steve Pennington, who's certainly a huge support in all of these studies and work. So thank you very much, everybody.
We have time for questions. Thank you for a great talk. But questions?
What I see commonly is morning stiffness without frank arthritis or enthesitis. You put that, are you a lumper? Do you call that psoriatic arthritis, or do you? To me, it's part of the spectrum, psoriatic arthritis. So
Yeah.
it responds to therapy as well.
Yeah.
Talk a little bit about your ideas about morning stiffness.
Yeah. Well, morning stiffness and joint pain are certainly some of the features that patients with evolving, if you like, psoriatic arthritis may experience, and they may experience it for years before they eventually develop anything that you can see. So those patients may well be on their way to psoriatic arthritis. There are, of course, other musculoskeletal problems that can occur in the same population, and you do have to be careful to differentiate it from commonly things like osteoarthritis, rheumatoid arthritis, and other forms of disease. But yeah, many of those people are evolving, and certainly the work from Toronto would suggest that stiffness and arthralgia are the key symptoms to look out for in your patients who are likely to go that way.
Great.
Screening for asymptomatic enthesitis will help diagnose psoriatic arthritis. Are they included?
Screening them how?
Screening them just like doing ultrasound of the enthesitis.
Yeah. It may be helpful, and but the ultrasound is certainly very much user dependent, so it'll depend on the skill and the expertise of the person who's doing the ultrasound. There also are many enthesial sites in the body, So you have to decide which endothelial sites you're going to choose.
For psoriasis, I think they usually go for 6 endothelial sites.
Yeah, yeah, yeah. I think it will pick up some people, yes. I don't know that it would. I don't know that it's the future, to be honest. I think it's very labour dependent, and I'm very hopeful that we will find something that's a little bit easier to apply to a lot more people than having to do an ultrasound of all their endothelial sites, or even 6 sites would be difficult.
I got one question for
Sure.
you.
There's one at the back as well.
I'll let you go first.
For your 3 groups of the psoriatic arthritis, have there been any studies about a therapeutic response for which therapy may be better for which may be?
Formal studies, no, but there, I suppose the study mentioned earlier relating to HLA-CO6 positivity and its response to ustekinumab or adalimumab would suggest that those patients may be responding differently. I think that study yet needs to be done. I think it's an important study. I think the pharmaceutical companies will have a lot of those data. They've done HLA typing on a lot of the patients and would be able to do more of that analysis, but hasn't been done yet.
Well, one quick question. Maybe not an easy one, but so we clearly have 2, closely associated diseases: psoriasis and psoriatic arthritis, both inflammatory, genetically determined. Clearly with a key role for CD8 positive T cells and IL-17. Any insight into why there is this lag time between the psoriasis and typically the development of psoriatic arthritis? Is their onset completely unrelated or disconnected? Or is there a relationship between the 2?
Yeah, I mean, certainly there appears to be a difference in lag time depending on what HLA type you have. How that relates to a CD8 positive T cell, I'm not sure that I understand that mechanism or what the explanation for the lag time. It's a very good question. I don't know if I know the answer to that. Have you a suggestion yourself?
I have no idea. So
Often I find that if people ask us difficult questions, they know the answer.
no, we're on the same page. So thank you. I think.
Thank you very much.
Great talk. Thank you.